So this section or this chapter is on the chemical levels of organization. We will learn the role of chemistry in our lives. There are several questions that come to our mind when in our day-to-day activities. For example, why is the sodium or cholesterol levels when high or low harmful to the body? Why, when you have diarrhea or you're vomiting, it can be detrimental to your body? What is anemia? What is iron deficiency anemia? These are the several questions that come to our mind, and we have to face them in our day-to-day activities. For this, we need to know the chemical levels of organization. So first, let us understand the role of chemistry in the life. Okay, chemistry is a science that deals with structure of matter. Okay, so the question is, what is matter? Matter is defined as something that has mass and has space. Okay, that occupies a space, that has mass and occupies some space, is known as matter. What is mass? Mass is the amount of material in matter is considered to be mass. So what is the amount of material in a typical substance is known as mass. So matter actually exists in three different forms, or three states of matter exists. Number one, solid, liquid, and gas. So all matters are in this form, either in solid form, liquid, or gaseous form. So let's understand now. So all of these, the different substances around us, okay, we are made up of different substances which are called elements. Okay, so these elements actually are unique substances that cannot be broken down by ordinary chemical means. So you emphasize on the word ordinary because eventually we will learn how these different elements can be broken down. But by ordinary chemical means, these are unique substances that are present around us. These are, the matter is made up of these elements. Now, atoms, these are the building blocks of the elements. So atoms make up the elements. The atoms, there are many atoms in at present. These are the smallest stable units of matter is atoms, and they are the building blocks of the element. Air, whether it's elephant, oranges, oceans, rocks, and people, are all composed of atoms in different combinations. The unique characteristics of each object, living, non-living, result from the different types of atoms involved and the ways these atoms combine and interact. And these atoms have atomic symbols, and we learn that eventually. So we have some major elements in our body. There are many other elements, but I just want to make sure that you understand that there are 91 natural occurring elements on the earth, and 24 of which play normal physiological roles in human. In this chart, as you can see it in here, the top of the ladder is oxygen, and it occupies 65% of human body weight. Carbon, which is denoted by C, occupies about 19%. Hydrogen, which is denoted by H, occupies about 10%. So the top three, you do have to remember: oxygen, carbon, and hydrogen, because eventually we will see how important these elements are in our body. So atoms, these elements are made up of atoms. Atoms have a general structure. They are made up of three subatomic particles. Number 1, protons. Number 2, neutrons. And number 3, electrons. And we learn each one of them. Number 1 is, let's understand protons. These are positively charged and have mass. Neutrons have no charge and have mass. Electrons are negatively charged and have negligible mass, or you can say they have a mass of 1 by 2,000th the mass of a proton, which is negligible. Again, protons are positively charged and they have mass. Neutrons have no charge but they have mass. Electrons are negatively charged and they have negligible amount of mass. So if you look in here, in the center are present the protons and the neutrons. Again, protons are positively charged, neutrons are neutral, and revolving around them is the electrons. I want you to understand. So the center part, which consists of protons and neutrons, is called the nucleus. And the electrons revolve around the nucleus. And these electrons revolving around the nucleus, these orbits are known as either orbits, they can be called orbitals, they can be called electronic shells. Okay, so you have to understand there are different names for it. So orbits, orbitals, electronic shells, valence shells, there's the fourth one, valence shells. These are all different names in which these electrons revolve around the nucleus. The structure, the entire structure, which consists of protons, neutrons, and electrons, scientists came up with different models as to how they are organized. Number 1 is called the planetary model, which looks like the solar system with the sun in the center and the planets revolving around it. So you can see this is the center part, and this is where the electrons are basically the planets which revolve around the sun. As days went on, it became more clear that these electronic shells are not so discrete. They are not so discrete, and most likely they are regions around which they revolve. So then came the concept of orbital models. So these shells, or these electronic shells, were then considered that it's not one orbit, there are many orbits on which the electrons move, and it is more like a cloud than a really straight line in which the electrons would move. Okay. Some terms that you need to know. One is the atomic number. So what is atomic number? It is equal to the number of protons that is present in an atom. Okay, so if the atomic number of an atom is 6, means the number of proton is 6. Okay, so I want you to understand this concept very well, is that the number of proton is equal to the number of electrons. Why? Because atom is electrically neutral. So the number of positive charge has to be equal to the number of negatively charged. What is number of positively charged? It's protons. So number of negatively charged is electrons. So this has to balance each other. So if the atomic number of an atom is 6, the number of proton is 6, number of electron is also 6. Okay, so that makes it clear. Always remember again, atom is electrically neutral. Next comes the mass number. This is equal to the mass of proton and neutron. So basically, if we see it, if we calculate the mass number, it is the mass of the protons and mass of the neutrons. Then comes isotopes. What are isotopes? Atoms with the same number of protons but have different number of neutrons. So here, please understand, protons are positively charged, neutrons are neutral. So isotopes have the same charge. So a number of protons are the same, but the number of neutrons are different. So we will learn about the different elements, different atoms with their atomic number in the first slide, and then we learn about the isotope in the subsequent slide. So these are the different kinds of elements. Number one is shown hydrogen, helium, and lithium. So hydrogen has 1 proton, 0 neutron, and 1 electron. So as you can see it in here, the atomic number of this atom is 1. Why? Because the number of proton is 1. I want to reiterate what I just said before. Atom is electrically neutral, so number of protons is equal to the number of electrons. So it is electrically neutral. So atomic number is 1. Okay, helium. Helium, 2 protons, 2 neutrons, and 2 electrons. So the atomic number is 2, and you can see the number of protons are true, and number of electrons also is true, 2. Then comes lithium. Lithium has 3 protons, 4 neutrons, and 3 electrons again. So again, atomic number is 3, number of protons 3, number of electrons is 3. In here, what is shown is that the number of neutrons in each of these cases are different. So atoms can have same number of protons and electrons. If they have a different number of neutrons, they are a different atom, or they form a different element eventually. Okay. So here is shown isotopes, okay? This is the isotopes of hydrogen. Hydrogen, deuterium, and tritium. You have to remember, for isotopes, the number of, or atomic number is the same. So for example, in hydrogen, the atomic number is 1. In deuterium, atomic number is 1. Tritium, atomic number is 1. What does that mean? It means number of proton is 1. Number of electron is 1. For deuterium, number of proton is 1, number of electron is 1. Tritium, number of proton is 1, number of electron is 1. But they are isotopes of each other. That means the number of neutrons, if you see, is different among them. Okay, so number of neutrons in case of isotopes are different. I'm going back in slide to show you one similar thing. In here, number of protons, neutrons were both different in each of these elements. So these are not isotopes of each other, but these elements who have the same atomic number are isotopes of each other. Now, now we need to understand something, which is that these atoms have different atomic number, and some are more stable than the others. Okay, some have more stability or control on the number of electrons and neutrons than others. Here specifically, we are interested in number of electrons. The electrons are arranged in different shells. If you remember, we just learned how these are called orbits, orbitals, electronic shells. These electronic shells, arrangement of these electrons are different in different elements depending upon the number of electrons it has. The first electronic shell always holds 2 electrons. The next electronic shell holds 8. The third electronic shell holds 18. So 2, 8, 18, or it can be 2, 8, 8. So in each of these cases, if the octave or the electronic shell has 2 electrons, then the atom is called inert because it is very stable and it will not go into any chemical bonding. If the atom, on the other hand, has 1 electron, then it is very chemically active and it will try to go for bonding. So any atom that has its first shell 2 electrons, that's considered to be very stable. Another atom which has a first shell 2 and the second shell 8 is again very chemically inert and will not go into bonding and is known as inert element. Again, the third element, which can be 2, 8, and 8 or 2, 8, and 18, they are also very chemically stable and are known as inert elements. Any other elements which have lower than the complete octave of 2, 8, 18 or 2, 8, 8 are chemically active and will go into what is called bonding. Okay. So if you look in here, so in this case, you can see hydrogen has 1 electron in the outermost shell. As such, it is very chemically active. The other is carbon. It has first shell 2 and the next shell it has 4. Again, it is very chemically active. Oxygen has 2 and 6. Sodium has 2, 8, and 1. And in each of these cases, you can see that the shells are not completed with the electrons that it can hold. As such, they go into chemical bonding. So the chemical bonding is of 3 types. Number 1, it is ionic, chemical, and hydrogen. Sorry again, ionic, covalent, and hydrogen. These are the 3 different kinds of chemical bonding. Ionic bonding, okay? Ions are formed in this case, okay? What does that mean? It means that electrons are transferred from one atom to the other in order to go into stability. So they are going to form a bond. That means they are going to either give away some electrons to form stability, or they are going to take up some electrons in order to gain stability. Or stability or stable environment, what it means is it is trying to achieve the inert compounds configuration, which means that it is trying to achieve 2 electrons in its first shell, or it is trying to get 2 plus 8 electrons in the second shell, or it is trying to get 2, 8, 18 or 2, 8, 8 on its electronic shell so that it forms the configuration of an inert compound or inert gas, it is said. So 2 atoms that gain 1 or more electron is known as anions, and atoms that lose 1 or more electrons are known as cations. So they form ions, anions, and cations. So what are anions? That have gained 1 or more electrons. So when they gain 1 or more electrons, it results in a more negative charge. Cations lose electrons, so they give away negative charge. As such, they become positively charged. So anions are negatively charged, cations are positively charged. So let's understand how it takes place, the ionic bonding. Ionic bonds form between atoms by transfer of 1 or more electrons. When they transfer electrons, when they give away electrons, as I said, they become positively charged, and the atom that receives electron becomes negatively charged. The best example is sodium chloride in NaCl. And NaCl has an electronic atomic number of 11. So the distribution of electron is 2 plus 8 plus 1. Okay, so you can see it in here, 2 plus 8 plus 1. On the other hand, chlorine has atomic number 17. The electronic configuration is first shell 2, second shell 8, and third shell 7. Okay, so I want you to understand, for sodium there is 1 extra electron on the third shell, and chlorine has 7 electrons on the third shell. In order to gain stability, sodium will try to give away 1 electron. On the other hand, chlorine will try to take up an electron. In that case, this transfer of electron from one to the other, or gain of electron from one to the other, is known as an ionic bond. As a result, you can see there is an ionic bond formed, but there are charges that are formed as a result of it. Sodium gets a positively charged because it gives away 1 electron. Chloride, it gains 1 electron. As a result, it becomes negatively charged, and there forms a bond which is an ionic bond, and the compound form is called NaCl, which is common salt. Next comes the covalent bond. Covalent bond is more like sharing of electron. In this case, atoms don't give up electrons, atoms don't take up electrons, they share in order to achieve the inert gas configuration. Okay, share means, for example, you have $500 and your friend has $500, and you rent a room which is $1,000 and you share it. Okay, so you're sharing the property amongst yourself. Okay, and this, by doing that, both are gaining something, and that is nothing but covalent bonding. In this case, atoms are sharing electrons amongst themselves. I point here, they do not give up electrons, they are just sharing their electrons with one another. So let's take the example of methane gas, which is the formula is CH4. So this is 1 carbon and 4 hydrogen. So this is carbon, which has an electronic configuration of 6, which is atomic number 6. So 2 atoms in the first shell and 4 atoms in the second shell. 2 in the first shell, 4 in the other shell. So carbon, which has atomic number 6, 2 plus 4 is the electronic configuration. So in order to achieve the inert configuration, carbon requires 4 additional hydrogen. Or what it can do, it can give up the 4 electrons that is on the outermost shell. Both are difficult. So what it does, in order to instead of giving up 4 electrons, it shares the electron with 4 different hydrogen atoms. If you remember, hydrogen has atomic number 1, so it has 1 electron on its electronic shell. So each of these 4 electrons will be sharing the electron with 4 other hydrogen. As a result, they form a bond and then forms a compound or a molecule, you can say, which is called the methane gas, CH4, and it's written this way. We have several such compounds, as we will discuss eventually, but I want you to understand this concept that in this case there is no charge form per se. Okay, right now what we are learning is we are not seeing any charge form, only there is sharing of electrons, and that is equal sharing of electrons. Now, this electronic sharing can be of different levels. One is it can form a single bond, or it can form a double bond, or it can form a triple bond. So the first is when it is forming a single bond. In case of methane, 1 electron from carbon, 1 electron from hydrogen, they're forming a bond, and as such this is a single bond covalent bonding that is formed in here. In case of oxygen, it is little different. The first shell is 2 electron. The second shell has 6 electrons. So it requires 2 electrons from the other compound or other atom. In this case, it shares its electron with another oxygen, which also requires 2 electrons. As such, they form a bond, a covalent bond with 2 of its electrons. So now oxygen has 2 electrons in the first shell and has 6 electrons of its own and 2 electrons from the other oxygen. So it has 8 electrons on the second shell. So this is O2 or oxygen gas that is formed. The triple bond can be formed exactly the same way. Nitrogen has 2 electrons in this first shell, has 5 electrons on the third shell. It requires 3 more electrons, so it can share with another nitrogen to form a triple bond. And as I said, these are not charged because they are having equal amount of sharing of electrons. Now we need to understand what we learned so far is equal sharing of electrons. Now, there can be sharing of electrons, but it can be unequal sharing of electrons, okay? When there is equal sharing of electrons, there is no charge formed, and it is called a non-polar molecules, okay? When there is unequal sharing of electron, it is called polar molecules because it develops a slight positive or slight negative charge known as delta positive or delta negative. Okay, so we'll learn about that, but just remember, I'll go back to the example that I gave. If we are renting a room, 500 given by X person, 500 given by Y person, and they share the room, that is a non-polar molecule because there is equal sharing of molecules, electrons. Now, if X gives $600 and Y gives $400, you still rent the apartment, but the amount of money that is given or shared is unequal. Same thing in here, unequal sharing of electrons. Some atoms have more attraction towards electrons and some have less. As a result, it results in an unequal sharing of electrons. So I want to give you an example in which we will come to hydrogen bonding. But remember, in water, H2O, what happens is oxygen and hydrogen are formed. So the example is in here. So we will go back to the polar covalent bond and then I'll come to the hydrogen bond. So if you see, this is oxygen and this is hydrogen. Oxygen requires 2 electrons in order to complete its octave. That is 2 plus 6 it needs additional 2. So as a result, it bonds with 2 hydrogen atoms. Hydrogen requires 1 electron, so it will, bond with oxygen. But since oxygen has 6 electrons on its outermost shell, it has more power, you can say. It has more control on its electrons. As a result, it develops a delta negative charge and hydrogen develops a delta positive charge. So this is a covalent bond, but it is a polar covalent bond when there is sharing, but the sharing of electrons is unequal. This leads us to the next bonding, which is known as the hydrogen bonding. The hydrogen bonding is a very weak kind of bonding. You have to remember that. It's a very weak. Covalent bonding was the strongest kind of bonding. Hydrogen bonding is a weak attraction between slightly positive hydrogen atom in one molecule and a slightly negative oxygen on the other. So if you see in here, the oxygen molecule has slightly negative charge and the hydrogen molecule has slightly positively charged. And these two molecules are they come together to form a bond which is known as the hydrogen bond. Water, as you can see it in here, water is the best example. So each of these molecules are held together by these hydrogen bond which is present in here. Hydrogen bond is also present in several proteins and DNA, where it is very useful to form coils. It forms three-dimensional structure, although it's a weak bond, but it has many, many functions. This leads us to the second part of the lecture, which is biochemistry. Biochemistry. So we are going to learn about the compounds with which our body is made up of. And they are classified into two parts, organic compounds and inorganic compounds. Organic compounds always contain elements carbon and hydrogen and are generally also accompanied with oxygen. So carbon, hydrogen, and oxygen are the main elements in the organic compounds. There are other elements also, but necessarily carbon, hydrogen, and oxygen is present. In inorganic compounds, on the other hand, carbon and hydrogen is not present. There are exceptions to that rule, of course, like water, which is H2O, has hydrogen and oxygen, but it is an inorganic compound. CO2, which has carbon, is an inorganic compound. But there are exceptions, but in general, inorganic compounds do not have carbon and hydrogen. So we are going to start with the inorganic compounds first, and then we'll go into the organic compounds. So let's understand the different chemicals reaction that take place, and then we'll come into the inorganic compounds involving them. Cells stay alive and function by controlling chemical reactions. In a chemical reaction, new chemical bonds are made, they are broken, they are remade, and so on. These changes take place as atoms in the reacting substance called reactants, and then they produce what is called a new compound or a different compound, which is called the products. So as you can see in here, in this slide, it's shown that there are chemical bonds that are formed, rearranged, or broken. And these different chemical compounds arranged in the form of reactants and products. So here is shown the example, hydrogen. Two of these hydrogen atoms, which are known as reactants, they form the hydrogen gas, which is a product. So these changes as the atoms are reacting, which are known as reactants, they are rearranged to form different substances, which is the product. So here on, let's understand what are the different parameters that involve or help or influence the rate of chemical reaction. Temperature. With increase in the temperature, usually the chemical reaction always proceeds faster. Particle size. The smaller the particle size, the faster usually the chemical reaction is. Larger molecules require much higher time or require more temperature in order to do the reaction. So particle size, smaller the size, faster is the chemical reaction. Concentration. Higher the concentration, faster is the chemical reaction. Catalyst, important. They are chemicals that increase the rate of reaction without themselves being changed. So these catalysts are responsible to increase the rate of reaction. Enzymes in our body are biological catalysts, and they help to increase the rate of reaction in our body. Now, there are several reactions that take place in our body, and these reactions are a combination reactions. They can be decomposition reaction or many other forms of it, but they are in general classified into three parts. Number one, combination reaction or synthesis reaction. Number two, decomposition reaction. And number three, exchange reaction. The first one, which is the combination reaction or synthesis reaction, is basically in which there is assembly of smaller molecules together to form a larger molecule. For example, you can see A and B join together to form AB. So an example of it would be in our body are different amino acids, they join together to form a protein. Okay, so this is an example of combination reaction where two elements or two substances join together to form a product. Next comes the decomposition reaction, which breaks a molecule into smaller fragments. Molecules, large molecules, are broken down into smaller molecules. And this actually is absolutely necessary in order to facilitate, for example, digestion. Larger molecules, it can be polysaccharides, disaccharides, are dissolved in water and they are broken into smaller glucose molecule in order to facilitate the reaction. So AB, which is a large molecule, is broken down by A and B. Now the next is the exchange reaction. Parts of the reacting molecules are shuffled around to produce new products. For example, AB reacts with C to form AC plus B. For example, again, glucose in the presence of water forms CO2 and H2O. So there is shuffling of the reacting molecules to form the products. So these these reactions are continuously going on in your body, and they are absolutely necessary in order for you to function properly. So the question now arises that there is the regulation, the fluids or the body fluids, how are they regulated? Regulation of the body fluid is done by the pH, which is absolutely vital for homeostasis. So before we even understand pH, let's understand a little bit brief of the background behind pH. We have learned that hydrogen atom has atomic number 1, if you remember. So it has 1 electron on its outer shell. This makes hydrogen very, very easy to go into chemical bonding. That is, it can easily give up 1 electron to form hydrogen plus ion or hydrogen ion. A hydrogen atom involved in a chemical bond or participating in a chemical reaction can easily lose an electron to become hydrogen ion. Hydrogen ions are extremely reactive in solution. In excessive numbers, remember, they can break down chemical reaction, change the shape of complex molecules, and generally disrupt the cell and tissue function. As a result, the concentration of hydrogen atoms in the body must be highly regulated. And there comes the importance of the pH. So what is pH? pH is the hydrogen ion concentration in body fluids. That is pH, but mathematically it is a negative log of hydrogen ion concentration. The pH of a solution is defined as the negative logarithm of hydrogen ion concentration and is measured in moles per liter. It ranges from 0 to 14, as you can see. This is a pH scale by which we can measure the hydrogen ion concentration. The pH scale ranges from 0 to 14 with 7 in the middle, which is known as neutral. With lowering from 7 to maybe 0, this increases the acidity of the solution. With increase in the pH, it increases the basicity or alkalinity of the solution. Okay, so let's take a little bit brief moment to understand what acidity and basicity is. So whenever in solution hydrogen ions are formed, a solution is dissociated to form hydrogen ion, that gives the acidic part of the solution. We know that's the acidity of an acid. Usually is a solute that actually dissolves in a solution to produce hydrogen ion. On the other hand, a base is a solution in a solute, is a solute which removes hydrogen ion from the solution, and usually that releases OH or hydroxyl ion. So going back, this is the neutral pH. If you go lower than 7, it becomes increasingly more acidic. If you go higher than 7 to 14, it becomes basic. That is, the hydroxyl ion concentration increases in here. This pH helps us to understand the acidity or basicity of the solution or solution in our body or fluids in our body. In the day-to-day lives, you can see these are the different substances you can measure the pH of a solution. For example, distilled water has pH 7, which is considered to be neutral. Coffee, which has increasing acidity, which is pH 7, is more acidic than distilled water. Same is true for lemon juice. Lemon juice, which has a pH of 2, is more acidic than coffee. On the other hand, if you consider household bleach, which pH 12, is definitely has a higher basicity than distilled water. Milk of magnesia and household bleach, if you consider these two, so household bleach has a higher basicity than milk of magnesia. So you understand, here we are talking about comparison between these different substances to understand the pH scale of our body. So too, this is in general in our day-to-day lives. In the body too, there are several fluids that require this pH regulation or hydrogen ion regulation in order to function better. So again, let's understand one last time. A solution with a pH 7 is said to be neutral because it contains equal number of hydrogen and hydroxyl ion. A solution is called acidic, meaning that it contains more hydrogen ions and less hydroxyl ion. A solution is considered to be basic or alkaline, meaning it has more hydroxyl ion than hydrogen ion. So this scale actually helps us to understand the different fluids in our body that have different pHs. This can be used in our day-to-day lives, in our bodies also. This takes us to acids and bases. And as we said, what are acids? An acid is a solute that dissolves in solution and releases hydrogen ion. So if you dissolve acid in a solution, usually water, it will give you hydrogen ion, thus lowering the pH. Remember, lowering the pH, it goes below 7. Because hydrogen ion atom because a hydrogen atom that loses its electron consists solely of a proton, right? If you remember, as I said, hydrogen has one electron and one electron is lost, so it just has one proton. Hydrogen ion are often referred to as, they are also called as proton donors. A strong acid dissolves completely in solution and a reaction occurs eventually, which is usually in one direction only. Usually hydrochloric acid, for example, will dissolve to form H plus and Cl minus, which is a strong acid. It ionizes, hydrochloric acid ionizes to form two ions. One is hydrogen ion, the other is chloride ion. Since it forms hydrogen ion in solution, it is an acid. A base is a solute usually which removes hydrogen ion from the solution and thereby acts as a proton acceptor. So hydrogen ion is a proton. Since it removes it from there, it is a proton acceptor. In solution, it usually produces hydroxyl ion. So if you see NaOH, sodium hydroxide, produces two ions, sodium ion and hydroxyl ion. So this hydroxyl ion usually is produced in case of bases. And this hydroxyl ion is able to bind with hydrogen ion to remove hydrogen ion from the solution. So bases actually act to release hydroxyl ion and are proton acceptors. Now, the other thing that we need to understand is the acid-base concentration, which we just learned, is the pH of an acidic solution and a basic solution, which is the acidic solution have higher hydrogen ion concentration, alkaline solution have a lower hydrogen ion solution, and neutral solution have equal number of hydrogen and hydroxyl solution. So before we go into this part, I just want to give you a little information regarding salts. We learned about acids, we learned about bases. Salts is another compound. A salt is an ionic compound consisting of any cation except hydrogen ion and any anion except hydroxyl ion. So basically, these are salt are, for example, NaCl dissociates in sodium and chloride. NaCl is a salt, okay? So this brings us to the next part, which is called buffers. Buffers are compound that stabilizes the pH of a solution by removing or replacing hydrogen ions. So they resist any change in the pH in the body fluids by stabilizing the pH of our body by either removing the hydrogen ion or replacing the hydrogen ion. For example, buffer systems involved in our body, a typical buffer system is sodium carbonic acid bicarbonate buffer system, as you can see it in here. The body's carbonic acid bicarbonate buffer, it consists of carbonic acid and sodium bicarbonate, NaHCO3, otherwise known as baking soda. Buffers and buffer systems in the body help to maintain the pH within normal limits. So these buffers are present in our body, which keeps the pH of our bodily fluids under normal conditions. For example, the human cell requires a pH of 7.4, as you can see it in here. And in order to keep that, there are several buffers that I said. The body's carbonic acid bicarbonate buffer system helps in maintaining the balance between them in order to keep the pH at 7.4. Now, we are moving on from the inorganic substances that we learned so far into organic substances. Organic substances are molecules that are present in the living system, which are primarily made up of carbon and hydrogen. They also have oxygen in them, and many can have sulfur, nitrogen, iron, and other substances, but they are rare. These organic substances are mainly of 4 different types. One is called carbohydrates, lipids, proteins, and nucleic acids. Again, carbohydrates, lipids, proteins, and nucleic acids. We'll go over each one of them as we go along. First, let's understand a little bit more about the organic compounds. So what does the word organic mean? Any molecule where carbon is bonded with hydrogen is known as organic molecule. There are several examples like methane, ethanol, formaldehyde, and glucose. These are all organic compounds. But many biological molecules are organic, like sugar molecule. Glucose is also a sugar. Steroids, proteins, DNA are all part of the organic molecule. So, organic compounds, as we just learned, always have carbon, hydrogen, and sometimes can have oxygen. They are usually covalently bonded with each other. I want to emphasize here one thing is that these organic compounds are made up of carbon and hydrogen, and rest all the other elements so far known are placed in the inorganic compounds. So there is definitely a unique property of carbon that gives it a completely separate group, which is the organic compounds. That is, carbon has a huge ability to form long chains. And these long chains with hydrogen or oxygen gives carbon a unique property to make different kinds of compounds. And that's coined as the organic compounds. 40% of the body mass are made up of these compounds, and there are 4 classes, as we just said. These are huge molecules, okay? These macromolecules are known as polymer, except for protein, which is not a polymer. Rest, all 3 are considered to be polymers because they are macromolecules. Some of the functional groups that are present in the organic compounds is carboxylic group, COOH. This is carboxylic group. And these carboxylic groups are present in fatty acids and amino acids. Amino group may be present, which is NH2, which is present in protein. The building blocks of proteins are amino acid, and it is present in the amino acids. Hydroxyl group may be present, OH. It is present in carbohydrates, amino acids, and fatty acids. Phosphate groups are rare, but present in some. Nucleic acid have phosphate groups, and phospholipids have phosphate groups. So these are, in general, these are the groups which each of these organic compounds are attached to, and they have unique properties that they play in our body. So starting with carbohydrates, I'm pretty sure all of you know what carbohydrates are, and you have carbohydrates every day. So let's understand a little bit more about carbohydrates. So proteins, all sugars, starches are made up of carbohydrates. So it is a very, very important source of energy and is stored in the body. Carbo and hydrate, these 2 words, carbo and hydrate. So it's made up of carbon and water. Water in turn is made of hydrogen and oxygen. So carbohydrates are made up of carbon, hydrogen, and oxygen. in the ratio of 1 is to 2 is to 1. So they are made in carbon, hydrogen, and oxygen in the ratio of 1 is to 2 is to 1. There are 3 different classes of carbohydrates: monosaccharides, disaccharides, and polysaccharides. Saccharides, these words, saccharides means sugar. Mono means 1. So if it has 1 sugar molecule, it's known as monosaccharide. If it has 2, it's known as disaccharide. When it has many, it is known as polysaccharides. So the main function of carbohydrate is energy. It stores energy in the form of ATP. It can store in the form of energy. It can store ATP in the form of energy in muscle, in liver. Sugar is also the building block of nucleic acid, which we will also be studying in this section. So some of the major functions of glucose is to provide energy. Usually it is in the form of glucose, which is present in the blood. Carbohydrates also are important because they can be present in the proteins as spark proteins, okay, and they can help in the building and maintaining of body tissue. Carbohydrates is a main source for regulating of nerve tissue or nerve cell, because glucose is the only source of energy that is taken up by the nerve cells. No other form of energy can be utilized by the nerve cells. Carbohydrates also have high fiber content. As such, they help in the process of constipation. They help to in certain diseases, to stop certain diseases like cancer, heart disease, and diabetes. So we are going to start with the structure of the carbohydrates. It is present carbon, hydrogen, and oxygen in the ratio of 1 is to 2 is to 1. It is classified into 3 parts. Monosaccharides, as you can see, glucose, fructose, galactose are all part of monosaccharide. Deoxyribose and ribose, which are the main sugars used in nucleotides or nucleic acids, are also monosaccharides. Disaccharides, when 2 monosaccharides join together by the dehydration process, releasing water, it forms a disaccharide. Sucrose is a combination of glucose and fructose. Maltose is a combination of 2 glucose molecules. Lactose is a combination of galactose and glucose. Lactose is present in milk. Polysaccharides, these are complex carbohydrates, okay? They are usually 10 to 100 monosaccharides. When they join together by dehydration process, they form complex carbohydrates. Starch, which is present in plants, and cellulose, which is also present in several animals. These are starch, cellulose are present, and they are important, have dietary, functions. They are important in our body, but these are complex carbohydrates and they are hard to digest. The starch is a glucose in plants, and high concentration of glucose in plants or animal cells is toxic. So surplus of glucose is converted to starch for safety. So whenever there is excess glucose, it can be stored in the form of starch so that it is not toxic to our body. Glycogen, again, glucose in our blood also. If you remember, glucose, if is in very high concentration in our blood, is taken up by insulin and converted into glycogen by the liver and stored so that as and when required, it can be released and can be used by the body. Cellulose is also called dietary fiber. Water soluble, found in fruits, veggies, and it is it's an insoluble fiber, is in nuts and grains also. So it does help in the movement of bowel and it is a complex carbohydrates. I want to mention here, complex carbohydrates is difficult to digest. So it needs a vigorous process in order to be broken down into smaller glucose molecules so that it can be digested by the body. Like carbohydrates, lipids, the word lip comes from lipos, L-I-P-O-S, called fat, contain hydrogen, carbon, hydrogen, and oxygen, okay? The carbon to hydrogen ratio is nearly 1:2. They have less oxygen than carbohydrates in general. So lipids are actually, lipids or fats, you can call them, have carbon, hydrogen, and oxygen, with oxygen being less in them, okay? They are hydrophobic, that is, they are insoluble in water, and they are classified into 5 different classes called fatty acids, eicosanoids, glycerides, steroids, and phospholipids. One of the most important function of lipids are that these are the structural components of the cell walls of the cell, okay? So phospholipids make up, it's called phospholipid bilayer because there are 2 layers of it, and they make up the cell wall. So it's absolutely necessary that we have lipids in our body. You might have heard that fats are not good for your health. Yes, in excessive amounts, they are not good for health, but they are required in our body. They're essential in our body because they are the building blocks of the cell wall. So some of the major functions of lipids, the second point we just discussed is they form the membranes along the cells, that is a cell wall. They store energy. They form hormones and vitamins. So lipids are essential for our body. So neutral fats, they are called triglycerides. As you can see, they are tri and then glyceride. So this is a glycerol molecule, this is the glycerol molecule where this glycerol molecule has 3 carbon atoms and hydroxyl atom attached to each one of them. And these hydroxyl atoms then have reactions with 3 sets of fatty acids. And these fatty acids have the carboxylic group, as you can see it in here. They react together to form what is known as a neutral fat or a triglyceride. Okay, so glycerol with 3 chains of fatty acid form a triglyceride. Okay, I want to mention here, if in case of 2 of these fatty acids or 1 of these fatty acids are replaced by a phosphate group, then it becomes phosphoglyceride. Okay, so when there are 3 fatty acids, it's triglyceride. When there are 2 fatty acids, it's diglyceride, and when one of the fatty acids is replaced by a phosphate group, it's known as phosphoglyceride. So the triglycerides can be classified actually into saturated and unsaturated fatty acids. Saturated fatty acids are found in many animal products and are solid at room temperature, and they are highly linked with the cardiovascular disease. So they are not very healthy form of fatty acids. Unsaturated fatty acids are liquid at room temperature and are usually have the plant origin. So we go into the next part, which is this just showing you how saturated fatty acids are solid. An example of it is beef fat. And peanut oil is an example of unsaturated fatty acid, which is liquid. There are other fatty acids, like, for example, phospholipids, steroids, and eicosanoids. If you remember, I just mentioned, phospholipids are the building blocks of the cell wall. What are phospholipids and their structure we'll learn in subsequent slide. First, remember they are modified glyceride and they have a phosphate group in them. The steroids, these are flat molecules with interlocking hydrogen rings. And iconosides, they are 20 carbon fatty acids, and they are also found in the cell walls, cell membranes, and example of them are prostaglandins. So phospholipid, as I was saying, phospholipids have a phosphate group and have two molecules of fatty chain, okay? So if you look in here, this is the glycerol molecule. As you can see, this is a glycerol molecule, and these are the two fatty chains to which it is attached to. And attached with it is a phosphate group here, okay? So that's the reason it's called phospholipid bilayer, phospholipids, and the phospholipid bilayer is for the cell wall, okay? So this forms a chain-like substance. So the glycerol black is the backbone of it. And there are two fatty chains. These are hydrophobic. This hydrophobic, the word phobic means phobia, means fear. Hydrophobic means they are fear for water, that is they are not soluble in water. And the phosphate part of it is polar head. It is hydrophilic. Philic means love. Hydrophilic means love for water. So they are soluble in water. So part of this is soluble in water, which is the polar head part. And part of it is insoluble in water, which is known as the fatty acid tail part, which is insoluble in water. So oils and waxes. Oils are very similar to polyunsaturated fatty acids. Oils are lipids that are liquid at room temperature. And oils we produce act of a natural sunblock, barrier to bacteria and lubricant, and act as lubricant for skin, okay? Waxes are lipids that are solid at room temperature. So oils and waxes are just solid and liquid parts of each. They are liquid, they are products that are, produced to protect dust particles from entering our body. So these waxes are produced so that some dust particles can be trapped in them and they don't enter into our main system of our human body. Steroids and iconosides. The steroids, remember, cholesterol is a steroid and absolutely necessary for our body, okay? Eggs have, cheese, milk have certain amount of cholesterol, and we need cholesterol because the cholesterol is the building blocks of many hormones, of many other lipids that are necessary for our body. There are these steroids are bile salts, vitamin D, sex hormones, estrogen and testosterone are all steroids, and there are adrenocortical hormones. So I'm going to go to the next slide and come back to this slide again. So if you see, cholesterol is the building block for testosterone formation and for estrogen formation in females. So going back to the last unit, which is the iconosides, lipid type derived from fatty acids is known as arachidonic acids, which is basically prostaglandins. So these are iconosides. Prostaglandin is an example of iconosides which are present in our body. Then comes proteins. Proteins, remember, are absolutely the most abundant organic compound in our body are proteins. They support, they have many functions, okay? They give support, they form the framework of, to provide strength on our body, they form a framework in the body. Collagen, keratin, etc. are all examples of it. Movement. There are several contractile proteins that are present in the muscle, which help in contraction and relaxation of the muscles. And these are nothing but proteins. There are transport proteins. Hemoglobin transports oxygen in blood. Hemoglobin is a protein. And that helps in the movement of oxygen. They basically hold the oxygen and carry it in the blood. Buffering, they help in assisting the pH changes. Albumin acts as an acid and base to prevent wide change in the pH in our body. So fluctuation in the pH is controlled by proteins also. Catalyst, several enzyme acts as catalyst, biocatalyst, to accelerate chemical reaction in cells. Regulation of metabolism, there are growth hormones, insulin, hormones in general are important for regulation or metabolism of our body, and those are all proteins. There are proteins which are involved in defense mechanism. Proteins called antibodies are components of the immune system, help to protect us from foreign matter. And there are several clotting factors which are also for the protective mechanism. They are involved to blood clotting. There are proteins in them which stop bleeding in the injury sites. They're also part of the defense mechanism. So proteins have, apart from being the structural framework in the body, they have many other functions also, as you can see it in here. So proteins actually is constructed by 20 amino acids, and they have a combination of carbon, hydrogen, oxygen, and nitrogen. So these are, they have a special structure and they are made up of amino acids, okay? And amino acid, as we just learned before, have an amine group and carboxylic group also. And we learn about the structure of proteins in the next slide, but just to let you know that proteins do have carbon, hydrogen, oxygen, and nitrogen, which is very important to remember. There are small proteins, there can be large proteins, small proteins in the range of 10 amino acids. It can be 2000 amino acids, which are larger, or macromolecules to form proteins. And the proteins have different structures. They can have primary structures, secondary, tertiary, and quaternary structures. And the shape of the protein actually determines the function of it, okay? How they form bonds, how they function depends upon the structure of the protein. And one of the most important example of it is hemoglobin, which we will learn. So the structure of proteins, the building blocks of protein are the amino acids. So if you look in here, so this is the general structure of a protein. So this is a carbon with its 4 bonds. So remember, these are the 4 electrons on the outermost shell, and they form bond with carboxylic group, which is the acid group in here. There is an amine group in here, which is NH2, and an H group, which is of course here, and there is an R group, which is a variable group. This variable group means that it changes from amino acid to amino acid. So basically rest of the group is same, it's only the R group that changes. So if it is H, it forms glycine. When it is CH2COOH, it forms aspartic acid. So the general structure remains the same, only the variable R group changes in the amino acid. Now these amino acids, they join together by the dehydration reaction, releasing a water molecule to form what is called a peptide bond, okay? So this peptide bond, when 2 amino acids join together, they form a bond which is known as the peptide bond, and many such amino acids join together via the peptide bond to form a protein, okay? So it can be 20, it can be 2000 such peptide bonds are formed, and it depends upon different kinds of amino acid that they join together to form those peptide bonds. Structurally, the proteins, as I said, has primary structure, secondary and tertiary and quaternary structure, and the structure of the protein determines the function of it. Usually the quaternary unit is present in hemoglobin, and hemoglobin actually is, sorry about that. And hemoglobin actually is, the 3-dimensional structure of protein is important for its function because hemoglobin carries oxygen to different parts of the body. So proteins, enzymes are proteins. So remember, all enzymes are not proteins. Okay, so all the enzymes are not proteins, but there are several enzymes which are proteins. Um, proteins are all proteins are not enzyme, but enzymes are many of the enzymes are proteins. So they act as catalysts for the reaction. Okay, so these enzymes usually end with the word ase, like catalase. So these enzymes are biocatalysts. They lower the activation energy. What is activation energy? I'll just go over it. But these enzyme actually increases the rate of reaction. So if you look in here, you'll understand it better. So I don't think I have that. So this protein in general actually act as to increase, they are responsible to increase the rate of reaction in a human body. So they are known as the biocatalysts. Comes to the last section, which is the nucleic acid. So the nucleic acids are a large organic molecule composed of carbon, hydrogen, oxygen, nitrogen, and phosphorus. So they have all of it: carbon, hydrogen, oxygen, nitrogen, and phosphorus. The two classes of nucleic acids are deoxyribonucleic acid, DNA, and RNA, which is a ribonucleic acid. So DNA is deoxyribonucleic acid and RNA is ribonucleic acid. So what is the primary role of these nucleic acid? The primary role of nucleic acid is storage and transfer of information. So they are mainly responsible for storing and delivering or transferring the information for the synthesis of protein. So from DNA, information goes to RNA, and from RNA, it makes protein. So it becomes the blueprint for protein formation. So, the nucleic acid consists of one or two long chains or subunits of nucleic acids are known as nucleotides. So it can be one nucleotide or it can be two nucleotides. And a molecule of DNA is a chain of nucleotides. So what is a nucleotide? A nucleotide is basically, it has a nitrogenous base, a pentose sugar, and a phosphate group. So let's understand what each one of them is, and we will learn about them in the next slide. But right here, I just want to show you, this is showing you a DNA. These are the nucleotides that are intertwined, double helix chain of nucleotides. And here are the composition of the DNA. So you can see it in here. The nucleotides are made up of nitrogenous bases. And it also has, you can see the nitrogenous bases it's made up of. It has pentose sugar and a phosphate group. It has a pentose sugar and has a phosphate group. We'll do in details in the next slide, okay, so that you understand it better. Okay. So nucleic acids are macromolecules, and they are made up of nucleotides we just learned. There are two kinds, DNA and RNA. DNA is deoxyribonucleic acid, RNA is ribonucleic acid, and they are made up of several substances. Let's do each one of them, okay? A typical nucleotide consists of a phosphate group, a sugar molecule, and a nitrogenous base, okay? So for example, there are the nitrogenous bases with one carbon nitrogen ring. So there is nitrogenous bases along with it, and they are as uracil, thymine, cytosine are called the pyrimidines, and purines are adenine and guanine. So they are of different kinds. So let's understand this once more. So they are, DNA and RNA is made up of phosphate group. They have a sugar molecule and they have nitrogenous bases, which is adenine, guanine, which are purines. Uracil, thymine, and cytosine are pyrimidines. So they are, these nucleotides are made up of nitrogenous base, which are of different kinds. Okay, so what are they made up of? They have phosphate group, they have a sugar moiety, and they have a nitrogenous group. So so for DNA, the sugar moiety is deoxyribose sugar, and for RNA it is the ribose sugar. So the ribose sugar in RNA, when it has one less oxygen in them, it forms deoxyribose sugar, and that's present, that sugar molecule is present in DNA. For RNA, the sugar molecule is ribose sugar. Both of them have phosphate group, and for DNA, the nitrogenous basis is ATGC, which is adenine, thymine, guanine, and cytosine. And for RNA, it is AUGC, which is adenine, uracil, thymine, adenine, uracil, guanine, and cytosine. AUGC. Okay, so these are the differences between the two nucleic acid. This actually shows you the nucleotide composition. This is the sugar moiety. This is the thymine, thymine. This is the nitrogenous base in here. It is attached to a phosphate molecule also. Okay, so these join together by double helix in order to form a nucleotide. So if you look in here, these are the bases, nitrogenous bases. This is cytosine, this is guanine. These are the phosphate molecule, and this is the ribose or the sugar molecule. So for DNA, it's a double helix, and these double helix nitrogenous bases join together by hydrogen bonding to form. They usually join as adenine joins with thymine, guanine joins with cytosine, ATGC, that's how they join together to form a double helix. They are intertwined with each other. RNA, on the other hand, the nucleotide is single nucleotide, and the bases are shown in here as AUGC. They have uracil instead of the thymine in DNA. This actually shows you adenine pairs with thymine, forming two stabilizing with hydrogen bonding. Guanine pairs with cytosine, forming the bond with hydrogen. RNA molecules in general, they contain ribose, not deoxyribose. They have uracil, they are single-stranded, and there are different types of them depending upon their function: messenger, ribosomal, and transfer RNA. Okay, ATP, adenosine triphosphate, is a high energy compound. ATP is required by muscles, by cells to do all the functions. It consists of 3 phosphate groups attached to adenine and 5 carbon sugar, ribose sugar. So this is an adenosine, and when it is joined to 3 phosphate, then it's known as adenosine triphosphate. So this is adenine, and this is the phosphate group. So when it has 3 phosphate group attached to it, it's known as adenosine triphosphate. When it has 2 phosphate, it is known as adenosine diphosphate. When it has 1 phosphate group attached to it, is known as adenosine monophosphate. And all 3 of them are involved in energy as an energy compound. But here we are more interested in ATP, which is adenosine triphosphate. And it is the main source of energy for cell for all its function. So these molecules, these macromolecules, are absolutely necessary for our body. The cell makes ATP by using sugar molecules in order to form ATP. There are other sources also, like amino acids, fatty acids can also form ATP to suffice the energy needs of the cell.